Good afternoon, everyone. My name is Ilan Chaitovitz, and I'm part of the team here at CRDN. And I'd just like to add to the warm welcome to all of you. It's great to see you here. Thanks for coming. I'm absolutely delighted to have the responsibility to introduce Professor Evan Reid, who, in my humble opinion, is one of the leading molecular biologists in the world. Although you probably wouldn't accept that.
Maybe in the top 1,000.
Well, listen to his bio and you can judge for yourselves. Evan, if I can call you that, graduated in medicine at Glasgow University in 1991. He subsequently completed his PhD in genetics and then did a stint as a Wellcome Senior Research Fellow. Since 2021, he has held the title of Professor of Neurogenetics and Molecular Neurobiology at Cambridge University. He is also a principal investigator at the Cambridge Institute for Medical Research and runs a specialized neurogenetics clinic at Addenbrooke's Hospital. Initially focusing on the genetic basis of hereditary spastic paraplegias, or HSPs for short, which is a degenerative motor neuron disease that affects around 50 people in a million. Evan's research now encompasses the entire spectrum of rare disease clinical features, underlying genetics, and cellular biology. With that, thank you.
Well, thanks very much for the kind introduction. I'm going to ask my mum to watch the YouTube video for this now because she'll lap all of that up. So I was given the task of talking to you a bit about if DNA is the blueprint, what is it the blueprint for? And Rishi in his in the first session this morning, I don't know if people were here for that. He basically gave the talk that I'm going to give, but he did it in musical form. So I can't promise to do it as entertainingly as that, but I'll do my best. So we're all here because of an interest in rare disease. And of course, while rare diseases are individually rare, collectively they're actually quite common. So we define in the UK a rare disease as affecting more than 1 in 2,000, or sorry, fewer than 1 in 2,000 of the population. So if you add that all together, you come up with really quite a large number of people. So we think there's probably something like 3.5 million people in the UK who are affected by some form of rare disease. And most of those conditions, or many of those conditions, have got a genetic basis. And particularly when you look at the pediatric spectrum of rare diseases, something like 80% of those have got a genetic basis. So it is really important that we understand DNA. And we previously had the session where we've talked a lot about practical applications of DNA sequencing. And again, in Rishi's talk, he gave us a great singing introduction to DNA. But I guess I just wanted to point out that DNA is this kind of 3 billion letter long DNA code. And here's the classical double helix structure of DNA that Rishi talked about before. And sometimes it's difficult to get the idea of just how colossal that code is. So, for example, if you were to say a letter of DNA in the genome once a second, if you started now and did that 24/7, it would take you something like 100 years to read through the whole genetic code. So it's a really colossal amount of information. But the key parts of the genetic information are really contained in the genes, and the genes make up only about 1 or 2% of that whole 3 billion letter DNA code. And when I started in genetics, which was longer ago than I sort of care to remember now, so probably about 1994, we used to say to families that we had something like 100,000 genes. And then, of course, the whole Human Genome Sequencing Project came along, and we realized it was nowhere near that amount, and then it went down to about 50,000, then 30,000. So we now think it's sort of roughly around about 20,000 genes that we have in total. And you can think of each gene as providing a blueprint for a part of the body. But if they're providing the blueprint, what's the builder and what are they building? So I can tell you in this case what they're building is the institute that I work in, so that's Cambridge Institute for Medical Research. But in the cells of the body, what's the builder and what's the building? So to do that, we need to think a little bit about how genes are working in cells. And again, Rishi sang a bit about this. But if you can think of the DNA code, which is in the nucleus of the cell, acting as the blueprint, what happens for the DNA code to work is essentially the DNA opens up, and then it's read, and an mRNA is formed, so that's the messenger. So you can think of the mRNA as being a bit like the courier that's taking the blueprint from the architect's office through to the builders. And then after that, what happens is the mRNA goes out into the exterior part of the cell, so outside the nucleus, and it makes these structures called ribosomes. And essentially, it's the ribosomes that are the builders. So you can think of ribosomes as basically being factories where the messenger RNA is read, and it's read off in sets of 3 letters. And as it's read by the ribosome, what happens is a protein chain is built. So essentially, it's the proteins that are the buildings. And the proteins start off as you can see here, as kind of linear structures. So they're just like bits of string. But in the cells of the body, what happens is those bits of string are folded into more complex structures. So again, like you can see here, the bit of string being folded into a much more complex knot. We can look at the different stages of that process and kind of look at them in a sequential fashion. So if you start off with the long bit of string that's initially made by the ribosome, then that can be formed into what we call secondary structures, which are things like helices or maybe pleats. And then those structures are then folded up into more complex structures. And then sometimes what happens is you'll get different proteins that come together to act together in concert. And here are some examples of some protein structures, so some common ones that people might have come across. So this is what the cystic fibrosis transmembrane receptor looks like. So that's the protein that's affected in cystic fibrosis. Here's hemoglobin, and you can see the hemoglobin's made up of 4 smaller molecules that come together and act in concert to do the function. And the little green parts here are the heme groups, and that's they contain iron, and that's where the oxygen is loaded onto hemoglobin. And then this is a structure of an antibody, and these bits here bind onto things that the body recognizes as non-self. So, for example, like the spike protein of a COVID virus. And then this is a kinesin. So kinesins are motor proteins that move things about within cells. And just to give you an idea of the scale of things, this is the length of the cystic fibrosis transmembrane receptor. So it's 6 millionths of a millimeter in length. So you can compare that to human hair that's maybe a millimeter, a couple of millimeters in diameter. So absolutely tiny structures. So I mentioned the proteins essentially you can regard as the buildings. But of course, buildings work in concert with other buildings. And so in a town like Cambridge, you'll have all sorts of different buildings doing different things. You know, you'll have the council headquarters, you'll have hospitals, railway stations, and so on. So what happens is that the buildings come together to make a bigger structure, a bigger functional structure. And then the cells of the body, essentially, the same sorts of things happen. So proteins come together with other things that I've labeled as other stuff. So they all come together to form the cells of the body. And altogether, we've got something like, we reckon, about 37 trillion cells. So again, a very big number. So you can see it written there with all of the zeros. And you know, there's just a few examples there of the types of cells that are present in the body. So you can see in the middle row there, these are white blood cells. Here's a neuron. That's a sperm cell. And these are cells that you might find in your gut. And each of those cells contains thousands of different proteins. So we can use techniques, for example, there's a technique called proteomics where we can figure out the whole repertoire of proteins within a cell. And when you do that in a nerve cell, you find that they've got something like 12,000 different types of protein within them. So here's a typical cell. And just to kind of orientate you, here's the nucleus of the cell. So that's the bit that contains the DNA. And you can see the cell's bounded by what we call a plasma membrane. So it's a kind of lipidy structure, a fatty structure, a bit like, you can think of it a bit like an egg yolk, the membrane of an egg yolk that outlines the cell. And then within the cell, there are lots of different substructures that we call organelles. And some proteins are located in organelles. The organelles also tend to be lined by lipid or fatty membranes. And so here's an example from my lab, done by a PhD student from a few years ago, Guy Pearson. And this is looking at a structure called the endoplasmic reticulum. So that forms a kind of lacy network within cells. And so to orientate you a bit, this is the nucleus of that cell, and here's in the cyan color here, the bluish color, is the endoplasmic reticulum. And what he's done is labeled a protein called Sec61 that we know is present on the endoplasmic reticulum. And then you can look with high-powered microscopes to see the structure of that. And we were interested in that because we know the structure of the endoplasmic reticulum is disrupted in some diseases. So some proteins kind of stick onto those cellular organelles and membranes. Others are not so much specifically located with organelles, but might be present throughout the cell, and maybe located in the kind of watery bits of the cell that are not membrane bound. So here's an example of microtubules. So microtubules are kind of long cable type structures that give the cell rigidity and shape. They act as a kind of scaffolding for the cell. And they also act as railways, so they're kind of multifunctional. They act as railways. And if you remember, I told you about a kinesin protein earlier on. The kinesin proteins walk along those railways and take cargoes with them and move them from one part of the cell to the other. And then this is just to give you an idea of how dynamic things are within cells. So here we've got a cell where we've labeled the endoplasmic reticulum that I mentioned before in red, and we've labeled another structure. These are actually structures called endosomes in the green. And this movie's taken over about a minute, so you can see how much movement there is going on inside cells at any time. So amazing to think that, you know, you've got 37 trillion cells in your body at the moment, and this sort of process is going on in probably pretty much all of them. So the take-home message for this part is essentially to say that proteins carry out thousands of functions within a cell. But what does this mean for genetic disease? So as Rishi nicely said, genetic diseases tend to happen when a spelling mistake, or what we call a mutation, occurs within a gene. And I told you that what genes are doing is essentially coding for proteins. So if you get a spelling mistake in the gene, you get an alteration in the protein. And that essentially causes a bad effect, and then that's what causes the disease. So most of the genetic diseases we deal with, it's this sort of process that's going on. Some genes don't code for proteins, but mostly the genes that cause genetic diseases are coding for proteins. So it's a bit like if you get your blueprint wrong, so you put your wall in the wrong place, that can have kind of fairly catastrophic effects on the building. So your building or your protein can come crashing down. So how do gene mistakes affect proteins then? So what I want to do is give you 3 examples of particular genetic conditions, just to give you a kind of flavour of how abnormalities in proteins can cause disease. And it's kind of simple when you think of it. You can have not enough of the protein or the activity of the protein. You can have too much of it, or its function can be mucked up in some way. And one of the ways that it can be mucked up is if the altered protein takes on some new function, so what we call a toxic gain of function of the protein. So the alteration in the protein kind of poisons the cell. So we'll look at each of those in turns. So the first example I want to talk about is when there's not enough protein made. So here what I want to do is tell you a bit about a condition called hereditary neuropathy with liability to pressure palsies, which is a bit of a mouthful, so we tend to abbreviate it to HNPP. And so I come from Glasgow and did my clinical training in Glasgow. And people in Glasgow, on the whole, this is a sweeping generalization, but they like to drink quite a lot. So in our receiving mornings, sometimes we would be seeing patients in our ward round who had too much to drink and got what we call in Glaswegian miraculous, and had maybe fallen asleep with their arm over a chair or something like that and spent the whole night like that. And they would wake up and then find that they couldn't move their wrist, so they had wrist drop. So that's something that can happen to any of us. But patients with HNPP, what happens is they get that type of problem, but on very minimal trauma or almost no trauma. So patients with HNPP tend to develop these kind of palsies, and the palsies will last 2 or 3 months, whereas if you don't have HNPP, typically they get back to normal quite quickly. And that's basically because that the PMP22 gene, which is the cause of this, so lack of the PMP222 gene, causes the myelin sheath of nerve cells to be affected. So the nerves that are affected in HNPP are these peripheral nerves that run from the spinal cord out to the muscles. And those have got very long projections that they send out, and the projections have got a kind of insulating substance around them called myelin, and PMP22 is one of the main components of myelin. So if you don't have enough of that, what happens is it makes the projection from the nerve cell very fragile, and so it makes the end of the nerves, what we call the axon, much more prone to degenerate. And you can see here, this is actually a nerve biopsy from a patient with HNP22, HNP22 loss. And you can see what's happened here is essentially the myelin sheath is very disrupted in those patients. So that's an example of loss of the gene or lack of the protein causing a disease. The other thing that you can have is too much normal activity. So a good example of that is the condition achondroplasia, which I guess we'd all recognize now. So here's a picture of Peter Dinklage, who's affected by achondroplasia. So it's a short stature condition. And the key problem in achondroplasia is in bone growth. So here's a picture of a tibia and fibula, so the lower part of the leg bones. And you can see here there's a kind of gap in the bone. And so that's a normal x-ray, if you like, from somebody who's growing. And that gap's made up of cartilaginous material, so that's kind of cartilage cells that are there. And that's actually the place where your bone grows. So those cartilage cells divide and grow, and then slowly what happens is they transform into bone, and that's the way that the bones lengthen and stretch. But in achondroplasia, what happens is you have an abnormality in a protein called FGFR3. So that stands for fibroblast growth factor receptor 3. And that's a protein that normally sits on the outside of the cell, on the plasma membrane. And you can think of it as being a bit of an outpost. So it's kind of scanning the outside of the cell and looking for particular signals that are telling the cartilage cells whether to keep growing or whether to stop growing. And so normally what it does is, for it to function, it likes to have a cuddle with another FGFR receptor. So 2 FGFR receptors come together, have a bit of an embrace like here. And when they do that, they get, like we do maybe, they get all excited and they start to kind of signal into the center of the cell. So it's like a flashing light at the exterior of the cell that basically says to the cartilage cell, stop growing. So and that tends to happen when they meet a molecule called FGF, so fibroblast growth factor. So essentially what this system's doing is turning off growth of this growth plate, this cartilage growth plate, as we call it here. Now, in patients who have got achondroplasia, what happens is there's an alteration in the protein that makes the 2 FGR proteins that come together, if you like, much more amorous, so they're much more likely to stick together, and they just continually signal the whole time. So essentially what they do is they switch off growth at the growth plate, and so that's why you get the short stature and short limbs in that particular condition. So basically the FGR3 receptors are just signaling continually rather than being switched off. And then a third example is where you'd have what we call a toxic gain of function in the protein. So a good example of that is a condition called Huntington's disease, which people may or may not have come across. It's not an uncommon neurogenetic disorder. So it's quite a nasty condition where people who are affected develop a dementia-type illness. They often have a movement disorder with it, so tics and chorea-type movements that we would call them. And it's caused by an expanded section in the Huntington's gene. So normally, in the Huntington's disease gene, there's an area of what we call a triplet repeat. So there are 3 letters of the gene that are just repeated. And typically, you know, I guess, hopefully if we went around this room and looked at the number of repeats in the Huntington's disease gene, you might find that most of us got something like 30 odd repeats. But if the number of repeats goes above a certain threshold, And we tend to think of that threshold as roughly about 37 repeats. You develop Huntington's disease. And that's because what happens is when you have an expanded repeat, you get an expanded section in the protein. So the repeat codes for an amino acid called glutamine. And so you get an expanded polyglutamine tract within the protein. And when that happens, that tract takes on new functions that the protein doesn't normally have. And it can do things like cause the protein to aggregate together, so clumps of the protein stick together, and that can have toxic effects on the cell. So here's one way in which that can affect the cell. So this is a cell nucleus that we're looking at, and the coloured structures here are the abnormal Huntington protein that's got the expanded section in it. And what happens is they form these kind of spiky aggregates within the nucleus. And those aggregates can poke into the membrane that lines the nucleus and poke holes in it. And that can cause things that are inside the nucleus to leak out. And we know that has a toxic effect, and it sort of increases inflammation within the cells. And it also can cause things outside the nucleus to leak into the nucleus. So that could be one way in which Huntington's disease is causing pathology. So that's a gain of a new function of the protein. So I've told you about, in a way, the bad things that can happen with DNA alterations, but there are some positives from it, in that when we study rare diseases, having knowledge of what the gene alteration is can give us a great way into understanding what the mechanisms are that are driving the gene, so understanding the types of mechanisms that I've just talked to you about. And the kind of model we use for that is that if we can understand the de-alterations that are causing a particular disease, then what we can do is we can model that disease in the laboratory in a dish. And we can do things like make brain organoids. So this is a mini brain in a dish. This is actually an image from a colleague of mine, Madeleine Lancaster, who works at the Laboratory of Molecular Biology in Cambridge. These mini brains in a dish have got the same type of structure in cortical organization and so on as the human brain. So they're a great way to kind of model neurogenetic disorders. And here are cells. These are human neurons again that we've modeled so that they are mimicking the hereditary spastic paraplegia condition that Ellen mentioned that I work on. And that can, once you model the disorders in a dish, then it leads to some cool things that you can do to try and really help with the disease. So you can start to understand the mechanisms. You can use the models for designing novel therapies and also testing novel therapies. So for example, you can look to see whether the drug that you think should treat the disease is affecting your models to rescue abnormalities that we see within the models. And it can also be used for developing better diagnostics for patients. And this is the sort of thing that we're very actively involved in CIMR. So the Institute's got around about 300 people who are involved in studying molecular mechanisms of disease in this way. So just to summarize, what I've told you is genes are the blueprints for proteins, and it's protein abnormalities that are the drivers of most genetic disease. And if we can study these abnormal proteins, it gives us a great way into understanding disease mechanisms, which hopefully can have beneficial effects in things like identifying new treatments, testing new treatments, and so on. But I think one thing we were talking about in the earlier session about being realistic about these things, that studying individual proteins is actually quite a tricky, difficult thing to do. And it takes a long time to really kind of deeply understand mechanisms of disease. And as I mentioned before, we've got 20,000 genes. That doesn't actually translate to 20,000 proteins because what can happen is one gene can produce several proteins or several variants of a protein. And so it actually means we probably have more like 100,000 different proteins and protein variants to study and understand the functions of all of those things. So that's quite difficult to scale at the moment. It's a kind of artisanal, bespoke industry in a way. Although there are methodologies coming along that, at least for some proteins and genetic conditions, there are more rapid ways that we can kind of understand the effects of abnormalities on the protein. And I just want to leave it at that, just to acknowledge funders of my work and people who provided some of the images that I've shown. So Guy Pearson, who was a PhD student in my lab, and Catherine Roger, who's a postdoc in my lab, now working for AstraZeneca. Yeah. Okay, thank you. And it just remains for me to say sometimes mRNAs and ribosomes produce some kind of quite interesting messages in the amino acids that they produce. So it just remains for me to say thank you for listening.
Thank you, Evan. Thank you so much. A real whistle-stop tour of genes to proteins to rare diseases. Thank you. We do have a few minutes for some questions. I don't know if there are any in the audience off the bat. Otherwise, I've got a burning question which I'd love to ask you, because Evan is obviously at the very, very cutting edge of science and hopefully leading to treatments in the near to medium term. So I'd love to know, what is the most exciting thing you've learned, say, over the last year, and that gives you some hope as to where things might lead going forward?
Yeah. I mean, there's a lot of things. I think maybe even going back further than the last year, what I'm constantly staggered by is the pace of advance in both genomics, which we've heard a lot about today. I mean, I started genetics, as we were saying, in 1994, something like that. And what we can do now in genomics is utterly science fiction, compared to our understanding of things in 1994. I never thought it would be possible that we could sequence an entire gene on the NHS for a patient. And, as you heard with the Next Generation Children's Study or the kind of rapid genome, I should say, as well as this, I'm a consultant clinical geneticist at Addenbrooke. So seeing patients with genetic conditions, and the idea that we could see a patient in the neonatal intensive care unit and have whole genome sequencing or whole exome sequencing results back within 3 weeks that you could then use for the benefits of those patients. I mean, that to me, that for myself sitting in 1994, that would have been science fiction. So that's when, so genomics had advanced really rapidly. But the other thing that really is dramatically advancing, well, there's a couple of things. One is stem cell technology. And actually the confluence of stem cell technology and precise genome editing is enormously powerful. So with stem cells, in terms of understanding diseases in the laboratory, what that means is, if you look, probably 10, 15 years ago, lots of the work that was done in human neurological disorders, you had to use, for example, mouse models to study those properly, because we couldn't really culture human neurons in a dish. But with stem cell technology, what that effectively means is that you could take a skin biopsy from somebody and grow the cells from the skin biopsy, and then you can turn those skin biopsy cells into pluripotent stem cells that you can then differentiate into pretty much any tissue of the body. And that includes neurons. So that gives us a great way of growing human neurons in a dish so that we can then study. So that's a great thing in itself. But when you add in the possibilities of precise genome editing to that, it gives you enormous possibilities. So I might go back a few slides. So these cells that you can see here, so those are human neurons in a dish. And they've been engineered so that we can go from stem cells to having those human neurons in around about 3 weeks or so. They're engineered so that they've got a switch within them, a molecular switch. If we put a drug onto those stem cells, they will differentiate into human neurons within 2 or 3 weeks. But they're also engineered to have CRISPR machinery within them. So for some people will know about CRISPR, but for people that don't know about CRISPR, it's a way in which you can modify the genome. So it's a molecule that you can express in cells that can do things like cut the genome. Increasingly, it can do things like make single letter changes within the genome. And what we've integrated into these cells is a flavor of CRISPR called CRISPR-I, which stands for CRISPR inhibition. So basically, what we can do is we can target that CRISPR-I machinery to any gene that we want and switch it off. And so we've done that for a whole range of the genes that are involved in hereditary spastic paraplegias. And then that allows us a great way to model them, compare them, look for similarities, look for differences, and try and understand the molecular mechanism. So those sorts of things are, while we're using it for the conditions I study, they can be applied to almost essentially any genetic disease to model the disease and understand it.
Fantastic, thank you. Well, unfortunately, we're come up against time, but I can't think of a better note to end on than how your research is looking and how it could, at some point in the years to come, lead to therapies for people with rare diseases. So thank you again, Professor Evan Reid. Thank you.
Thanks very much. Thank you.